Good morning, students. The objective of today's experiment is to study the floral features of China rose, which is Hibiscus rosa sinensis, and by the dissection of the flower, we'll study about the floral details. So, just for the procedure, we'll take a flower, full-grown hibiscus flower, and the description of the flower. About the description, just let me explain you. The flower is a terminal flower. At this flower, at the each and the end of the axis, a single flower is there. So the flower is a terminal flower. The flower is a pedicel. It is borne on a small stalk, which is the pedicel. The flower is pedicellate flower. The bracts are not present, so ebracteate condition. The flower is a hypogynous flower. Hypogynous flower like this. In this case, I have made it open. The floral structures, like which is the petals and sepals, they start just below the ovary. So when the floral structures are below the ovary, the condition is termed as hypogynous condition. Here epicalyx is present. These green leaf like structures which you can see, these are termed as the epicalyx. Just next to the this structure is again green leafy portion, which is the sepal. Single one is termed as sepal, and the group of sepals is termed as calyx. So this wall, the first wall of a flower, as epicalyx is not present in in every flower. In case of China rose, it is present. So the first wall of the flower is the calyx, group of sepals. In this case, you can see the sepals are fused together, they are joined together, so the condition is termed as gamosepalous condition. Next to this is a bright colored portion, which is the petals. Petals are free petals, they are not fused together, so the condition in this is polypetalous condition. Now about the estivation. Estivation means arrangement of sepals and petals with respect to each other. Here in the sepals, they are arranged end to end, since they are in fused condition, but they are arranged end to end, so the condition is valvate estivation. While in case of petals, here you can see that in the petals, the condition is that one side of the petal, just one side of the petal is above the the second one, and the one side is outside, one side inside, one side outside. So this condition is termed as twisted estivation. For the sepals, the condition is valvate, while for the petals, the condition is twisted estivation. Group of petals, you already know that the group of petals is the corolla. Then the female and male reproductive part. Female reproductive part is termed as androecium. In this case, these yellow portions, this one, the anther and the filament. Anther and filament together, it is termed as stamen. So many stamens are present on this stalk, and the androecium, male reproductive organ, the stamens are termed as androecium. The condition is monoadelphous, since the stamen are free and they are not fused together, they are free, the condition is monoadelphous condition. The stamens are epipetalous, they are above the petal or attached to the petal. As here you can see this portion, it starts with the petal, it is attached to the petal, so the condition is epipetalous condition. And epipetalous and basifixed, base from the basal portion, it is fixed, so basifixed and epipetalous condition. Now about the gynoecium. In the gynoecium, it is pentacarpellary. 5 carpels are present here. 5, 1, 2, 3, 4, 5 carpels are present. They are in fused condition, so the condition is syncarpous condition. The ovary is superior, that all the floral structures arise below the ovary. So the condition of ovary is superior locules. If we cut the ovary, there is 5 locules, so pentalocular. Placentation means when we cut the section in the next experiment, we will cut the section of the ovary. The presentation is axile presentation. Ovules are located at the axis. This is about the floral detail. Now when you have to dissect a flower, what you have to do now about the dissection process, just take a flower with the help of a needle. I'm not having needle at my home, so just with the help of a needle or a forceps, just make a single cut. Now just here, just you have to open the flower only without disturbing other floral structure. So with the help of a needle or a forceps, just open the flower, cut open the flower to show the floral detail. Here, this one, with the help of forceps I can. Now in this one style, just remove the, just be careful while doing this not to break the stigma or the style part of the, you have to just cut open the flower. After this, just take a piece of filter paper, make it moist, and on the filter paper you have to arrange the flower in such a manner that all the floral structure like this one, calyx, ovary, stigma, style, everything should be clear. For you I have already done one experiment, and this is like this. You have to arrange the flower in this manner, in this, on a moist filter paper I have arranged this, and you can see there is all the parts are clear, even the calyx, the ovary, style, stigma, everything is clear in this. So you have to show to the examiner in this manner all the floral details. So this is the experiment about the family Malvaceae. One more flower I'll show you about the ladyfinger flower in the next experiment. Thank you for today's class.